So our next speaker is Jeffrey Morris, and he's going to be speaking about summary of toxicology program for NRDA. Thank you.
Thank you. Thanks, everybody. Yeah, so I'm going to, kind of following with the theme today, I'll talk a little bit about what's come out and been disseminated to the public in one form or another for some of the toxicity testing that we were involved with, and many people in this room and at this meeting were also involved with, and how that's kind of come out to the public. And I'm going to touch on some of the maybe more interesting things that came out just from a science perspective. Here's our usual huge disclaimer. One thing I will point to is this website, and I can give people more details later if you want to see that. But this is basically the kind of the portal to the Programmatic Damage and Restoration Plan, the PDARP, which kind of outlines what happened during the case. In particular, what I'm going to talk about today is some of the work we did to support the injury assessment. And there's a lot of different facets to that, but in particular, kind of focusing on the toxicity testing program. So, give you a little bit overview of that program, touch on some interesting highlights and research outcomes, and talk a little bit about how that information is coming out into the kind of public forum through the PDARP and some other avenues. So, as far as the program overview, just kind of touch briefly on this, and we've presented on this, talked about it a lot, and there's other folks who are going to follow up and talk about it a little bit more, but try to give you a sense of what this program meant for toxicity testing in terms of toxicants, stressors, different species endpoints, and also want to mention our collaborators on this, and there's a lot more to it than just kind of a list of names. There's literally hundreds of people and dozens of labs that got involved with this. So just in a nutshell, this is a kind of a huge matrix of environmental toxicology in terms of the number of bioassays and other sorts of related chemical characterization tests that were conducted. So at this point, there's over 600 tests that were done over this 5 to 6 year period using 4 different oil samples that were collected during the spill, dispersant, and then those were used as contaminated sediments or in water and oil mixtures, water accommodated fractions. We also looked at surface slicks, and then many additional stressors. So in addition to the oil, we looked at the effects of UV light and temperature, dissolved oxygen, salinity, and added pathogens to see how that exacerbated the effects that you normally see just from oil exposure alone. In terms of species, and this is all information that's detailed in the PDARP, but we ended up testing over 21 fish species, 12 invertebrates, did a little bit of work with some surrogate reptiles for the turtle work, and also looked at some different bird species. This is all work that was done under the direction or in collaboration with NOAA, U.S. Fish and Wildlife Service, and the state of Louisiana. And then lots of different endpoints. Some that kind of normal, traditional tox endpoints like survival, growth, reproduction, but some more interesting ones too that looked at sort of sublethal effects or effects that potentially are sublethal until they're not, and they manifest in some kind of lethal outcome. So I'll touch on some of those as well, and I know that there's a lot of other people in other sessions and in this session that are going to kind of get into some of these in a little bit more detail, so I'll try to point that out when we get to those. In terms of collaborating laboratories, this slide doesn't really do justice, but it kind of gives you a sense of all the different labs that were involved in different places where testing was conducted. A lot of different folks along the Gulf, other universities around the country, other labs, both state and federal labs, universities, private labs, and then we did some work in different parts of the world because there's interesting species and interesting kind of aquaculture setups or very unique setups. So we did some work in Panama and also in Australia looking at tuna in those cases. So to kind of give you a little bit of a sense of what we were doing, these are the oils, and you've probably seen maybe this slide before, or at least some pictures of these different oils. But for the program, we focused on 4 different oils. One was an unweathered oil, and then we also artificially weather that oil slightly to just remove some of the more volatile compounds, mainly BTEX. And we had two oils, Slick A and Slick B, that were collected from the field, and those were skimmed off the surface, and they were all at different relative states of weathering. And so we kind of folded these in depending on the species and the exposure matrix that we were trying to look at with the program. There was a few different ways to do this in terms of the oil and water mixing. We used a range of different methods. One is a high energy blender method on the left there, where we mix the oil and water together in a blender and then let it settle for a short time and expose organisms to the waft that you get out of that. There's also some other methods that have been used previously, and we continued using those. One's a chemically enhanced water accommodated fraction, and that's the one in the middle. So basically you're mixing oil and water together, stirring it for about a day, and you're adding dispersant to that mix. And then we also looked at a low energy mixing method, which is the one on the right. And that's basically just putting some oil on top of the water and stirring it slowly. You can kind of see that may be kind of circling around there, but it's not fast enough energy that you actually create a vortex. So that one gives you a solution that mostly has some of the more soluble compounds in it. So you can imagine using these types of different methods along with the different oils, you can come up with a whole range of different concentrations and different sorts of mixtures to potentially test depending on the question you're asking. In addition to some of the water accommodated fractions, we also did a lot of sediment toxicity testing. And the sediment that we used for this, in some cases it was sediment that was collected from the field that was impacted by the deep water oil, or we also did a lot of work with spike sediments. We'd collect clean reference sediments from different areas and then add oil to that. And there's different ways to kind of expose organisms. This picture on the left here is some work that came out of Fern Galvez's lab at LSU, where we're looking at the impacts of suspended sediment on fungulus, killifish embryos. We also did some work at Auburn. This picture in the middle, it's kind of hard to see, but there's actually fiddler crabs in this. And this is looking at the effects of oil kind of coming to shore and settling on top of the sediment. We did some other work with University of Southern Mississippi, or some folks at the Sea Center down in Texas, where we're looking at exposing different types of benthic fish to sediments that have been mixed up with oil that we've collected from the field. So kind of give you that's a real brief example of some of the methods, and I've got a couple more slides I might get into a little bit more, but also kind of talking about research highlights. A lot of the slides that I have next are things that we literally pulled out of the PDARP, and there are, in addition to, and one thing I forgot to mention, the PDARP was written for kind of a broader audience. There's definitely a lot of technical information in there, but we definitely wrote it with a broader audience in mind. However, there are a lot of references in the PDARP to either things that have already been published by our group or others, and then we also kind of tried to back that up with a lot of more technical reports that are part of the administrative record. So some of what I'm going to go through is a combination of some of the information that's in the PDARP for kind of a broader audience, and then some of the more technical information as well. So touch on some of the water and sediment exposure. We'll talk a little bit about a few different aspects of this that turned out to be particularly important and also very interesting, including the photo-induced toxicity testing. So this is toxicity testing with UV light as an additional stressor. Looking at surface oil sheen toxicity, and then also some sublethal effects. And I put that sublethal in quotes because as folks will probably kind of get into a little bit more, when you're talking about cardiotoxicity, oftentimes you're assessing that on a live organism, but it's pretty well known that in many cases you could have a live organism and a couple days later it's going to be dead because of those sublethal effects. So you know, there are sublethal effects, and then there's also sort of delayed lethal effects. But I'll touch on a few of those and then kind of give you a little bit more information on where to find some of this information. So this is a slide out of the PDARP. This is kind of the summary slide on a lot of the waterborne toxicity. This is interesting, not particularly useful if you're interested in real technical details, because you can see that some of the ranges here that we're showing are pretty broad. So this is just kind of giving people a sense of where things fall in terms of sensitivity and the range of toxicity that we saw with different organisms. So what this is showing is the LC20, so this is concentration that kills at least 20% of the organisms exposed for some different plankton species, both fish and invertebrates. And you see some pretty wide ranges here. The x-axis is a sum of 50 PAHs that we measured in the WAFs. And the reason for some of the ranges on these is because, as an example with the mahi, the third one from the bottom, we show 96-hour embryo larval toxicity from anywhere from 1 to 40 micrograms per liter. And this is because the exposures that are done in this case use a lot of the different oils and mixing methods that I was describing earlier. So you kind of expect that because there's definitely some different toxicities associated with those different oils and how you mix them up. So this is an interesting slide, maybe not particularly useful depending on what details you're interested in, but all the details behind these numbers are included in technical reports. Same thing for sediment toxicity. There's some interesting findings here in terms of the TPH 50 concentrations that cause effects. You know, depending on where you're sampling in the field, a lot of times, just kind of give you some perspective, we saw what you maybe call background PAH levels in a lot of sediments in Louisiana that were probably down in maybe the 0.5 to 1 milligram per kilogram range, part per million, which is down in the lower end of what you see here. So there's a lot of areas that had contamination, maybe in the 1 to 10 range, and you definitely start to see effects for different sorts of critters at different sorts of endpoints, whether that's reduced growth or some kind of gill tissue damage or mortality. So I want to talk a little bit about photo-induced toxicity. This is a list of the 50 PAHs that we're typically summing, and there are at least 14 that are known to be photoactive. There's probably more than that. The red bars here give you kind of a sense of the ones in this profile that are known to be photoactive and potentially phototoxic. But the idea behind this is when you have some of these PAHs either in the water or taken up into the tissues in semi-transparent organisms, when UV light interacts with that, it creates reactive oxygen species that basically chew up the cell membranes and the tissues and cause a lot of damage very quickly. This is an example of some of the work that we highlighted in the PDARP. This is Fiddler crab Zoea, so these are larval fiddler crab tests. This is work done with Dr. Aaron Roberts' lab at University of North Texas, who conducted the bulk of all of the phototox testing. And this was done in collaboration with Dr. Jim Stuckel, who's at Auburn. But the idea behind this slide is to show you the difference. The three dose response curves here are all conducted with the same concentrations, but different levels of UV light, and you can see that that quickly starts to increase mortality. We took that and tried to do some modeling with it that was actually applied in the PDARP as well. So there's a lot behind this slide, but the basic idea is at depth, the UV light decreases, and so the toxicity potentially decreases a little bit. But the take-home message behind this slide is these are field samples, these green dots collected during the spill, and everything to the right of that is at least 20% toxic or more once you have UV light in the mix, and this would be for offshore areas. And we pretty consistently found that UV light increased that toxicity by 10 to 100 times. Getting to some surface oiling a little bit also. There was a huge area that was covered. I don't need to go into that anymore. We did some testing with sort of the thinnest possible sheen that we could generate, which was about 1 micron thick, to see how that affected different organisms. And there's a lot of toxicity associated with that. In this case, I'm showing you some results from some bay anchovy tests, and you can see the red line is when we're using a slick and exposing it to different levels of UV. The black line is a control, and you see that that red line on the far right is the actual average daily UV in the Gulf during the spill. So relevant concentrations, and it's something we can model and apply, and that is detailed in the PDARP as well. We also looked at cardiotoxicity, which I'm going to, in the interest of time, move through this a little bit more quickly. Johnny Cardona with NOAA is going to be talking about this in more detail on Thursday, but those ended up being very low concentrations that caused effects. Also, we did some work with the University of Miami. Most of the contingency down here, that's going to be Martin's going to be talking about this a little bit more, but another really interesting aspect of this was looking at effects on reduced swim performance. I do have a movie, but I think I'm out of time, and I know Martin's going to touch on this, so I'm going to... You want to see a movie? Okay.
Yeah, you got about...
The one thing anyone's interested in, right? He's going to skip that. And that won't play. Hold on.
Yeah.
Okay, so these are mahi-mahi looking down, and Martin will get into what this really means, but how to kind of quantify this. But these are basically treadmill tests for fish, and you can see the one on the top is a control, the one on the bottom was exposed to oil for 24 hours, and then they sequentially increase the speed that you're swimming these fish at, and you can see the one on the bottom has failed, has become exhausted. And so that's another quantification, and these effects occur at relatively low concentrations and relatively short durations. Another aspect of this that we looked at, which Natalie Ortel and Joe Griffith have a poster on, is looking at immune response effects. So oil exposure, in this case southern flounder, increase their susceptibility to natural pathogens. In this case, we had, we did a lot of work on this. This slide just kind of summarizes the effects of oil without a pathogen, no oil with a pathogen, and that oil pathogen combination over a relatively kind of a short-term exposure of about 7 days exposure to oil and a few hours exposure to a concentrated pathogen. And you see that we've seen this in other types of studies as well, that immune response is affected. Ryan Takashita has a poster. He's also with Apt Associates. He's going to get into this a little bit more on Thursday. But this is an attempt on our part to try to link up all these different sorts of modes of actions among multiple taxa. And it's kind of an interesting idea. This is a constellation because we couldn't do a flow chart. The arrows go every which way. But we see modes of action conserved across different taxa where you see similar effects on everything from fish and invertebrates to sea turtles and marine mammals. We're continuing this work, especially in some of the areas of phototoxicity, oil sheen toxicity, and then really trying to focus on data synthesis and modeling. And so to kind of recap where some of this information has either been released or is going to be available, there's a programmatic damage assessment restoration plan. There's also the administrative record, which has all these technical reports I was referring to. We're in the process of working with NOAA to finalize a comprehensive database that's going to be available to the public through NOAA's online data repository that's called Diver, which is in prep. Hopefully that'll be available in the coming months. And then there's been a lot of presentations at conferences like this in Gomery, lots of publications that have already come out and that are in the works, and dissertations and things like that have also kind of potentially got this information out. And then there's a lot more work to do. So we really are in kind of the data synthesis and reporting phase of this. And I'm out of time anyways, but that's me. And if we don't have time for questions, I'll be around so I can chat with people.
I got time for one question, about 30, 45 seconds.
I have a 30, 45 second question. This is a remarkable data set with every variable possible. However, when you look at the results you found and how applicable it is in what I call the real world, with sediment concentrations as low as total pH sediment content as low as 1 part per million, I don't know what to do with that because I have a concentration that already, if I was to try to clean up to, I start at Portland and dig down to Norfolk, and just take everything out. It just isn't useful to try to work with a responsible party, and I don't know what to do with it with a damage assessment because I'm already well above any kind of concentration that you now have in the literature that shows an effect. So I was wondering how you could help me with this.
Yeah, no, that's a good point. I think, like I said, these summary slides are a little bit useless in a way. It's interesting, but if you're actually trying to apply this, I think rather than think about it in terms of like a threshold and an LC20, you need to think about the whole response as a continuum and look at the dose response. If you've got concentrations that are already at 2 or 3, and you're looking at what does it mean if they're 5, 6, 10, 20, you can look at the entire data set for that study and look at effects above and beyond sort of what your background is, and not just focus on that one lowest concentration that happened to cause an issue in a clean environment. Cool. I'm getting pushed off.
No, it's fine. Thank you very much. We have one last speaker.